Hey everyone, welcome to my molecular biochemistry series, and in this episode we are going to focus on translation, which is synonymous with protein synthesis. And now previously I had talked about the concept of gene expression, which is DNA transcription, and we created a mature mRNA strand that is going to tell the ribosome what type of amino acids to link together and thus create proteins. Now, protein synthesis follows a three-step process with initiation, elongation, and then termination. And I am going to run through each of those steps, but before I do, if you haven't already, visit our website at piazzaresearch.org to get a better understanding of our mission, which is to understand our place in the universe. And to do that, we need to look towards the stars and see what life might be like elsewhere and how that changes our perspective of us. We also need to look at the microscopic level and the fabric of reality and everything in between: civilization, society, etc. So now we can jump into protein synthesis, and like I said, we had transcribed an mRNA strand, and it's going to bond with a 40S subunit of eukaryotic cells. In this scenario, if it was a prokaryote, it'd be the 30S subunit. And to distinguish between eukaryotes and prokaryotes, you can think of the E in eukaryotes as even, P as prime, because prokaryotes have ribosomes that have a 30-50, making up a 70S ribosome, and 3, 5, and 7 are prime, and then eukaryotes have 40-60, making up an 80. And so 4, 6, 8 are even numbers. So eukaryotic cells are going to have the RNA strand bonded with the 40S, and that's going to move in first, and then the 60S along with a tRNA with the T-arm tethered to the 60S unit is going to come in and begin the translation process. This is initiation, and usually it's going to begin with an AUG, which has a methionine beginning the translation process. It will be the first amino acid in a chain of amino acids. So then we have elongation taking place, and we are going to link more amino acids to incoming tRNAs. And this happens until you reach a stop codon, which would be either UGA, UAA, or UAG. And to remember that, you can use the mnemonic: you go away, you are away, or you are gone. And so the tRNA for the next amino acid comes in, and the elongation process begins at the A site, which is where the amino acyl tRNA is going to be. And then the P site is where you're going to lock in that tRNA within the ribosome. Now, at this point, you are going to forge the peptide bond between the methionine and the incoming amino acid, which could be anything or whatever the mRNA is coding for. And so once that peptide bond is solidified, the older tRNA is going to have no amino acid attached. It's going to be an empty tRNA and exit at the E site. Now we have this goes on and on and on until we reach a stop codon. And at that point, you have reached termination. GTP comes in, adds a phosphate, and you can remove the polypeptide chain. And you can do this multiple times, get multiple polypeptides. They can come together, coalesce, and form a quaternary structure. And then you have chaperones that may facilitate folding of the protein in various different manners, depending on what the cell needs and what it is programming the protein to do. And with that, we'll stop this video, and I'll see you guys in the next one. Take care.